electron flow through the two photosystems in the Z scheme. And as you might imagine, there was an incredible amount of oversimplification of what happens in electron transport between PS2 to PS1 to the final electron acceptor. And so where we're going to focus today is on water splitting and then electron flow from PS2 to PS1. Here is PS2, and associated with PS2 are extrinsic membrane proteins that are involved in water splitting, removal of electrons from water, and generation of a proton gradient. And as most of you remember, the proton gradient that is set up by electron transport is utilized for ATP synthesis. We'll talk about this at the end of the day. Electrons come from water splitting. P680 is our reaction center, and high energy photons transfer the energy through the antenna complex to our reaction center. We have two proteins in the reaction center, D1 and D2. Okay. What'll happen is that P680 gets excited with energy from a photon of light, and that excited electron is passed on to quinone. Okay. Now, one of the interesting characteristics of quinone is that it is a 2-electron acceptor. And so before plastic quinone leaves PS2, it's going to accept 2 electrons, and we call these 2 states, if it's accepted no electrons, it's QA, 1 electron is QB, and then 2 electrons is plastic quinone in this state. And at this point, plastic quinone is a small molecule that is mobile within the lipid portion of the lipid bilayer in our thylakoid membrane. Okay. Up is the stroma side of the chloroplast, and down in this one is the internal luminar space of the thylakoid. And as photosynthesis occurs, more and more protons are going to build up here. And we're going to see where these protons are coming from as we go through electron transport. So when you look at electron transport, remember not only how the electrons are moving, but also what are happening to the protons and where we're getting a proton gradient from.
So this entire thing is PS2?
This is just PS2.
Okay.
And this is kind of, so PS2 has 40 different proteins associated with it, okay? Some of those are simply chlorophyll AB binding proteins, so part of the antenna complex. And some of these are water splitting proteins, but this is a fairly large complex. And the reason that's important is that if electron transport goes awry, we're going to build up free radicals, and what it's going to do is to essentially interact with one of these proteins, oftentimes destroying it. So with very, very high light, oftentimes there are high levels of chaperones associated with protein refolding, and also high levels of proteases that are involved in the removal of proteins that have been destroyed, and then replacement and reassembly of the complex. So what's most important here is that electron flow is not disrupted, and we don't have any positive free radicals at any point through here. So D1 and D2 are the important proteins in our reaction centers. And the water splitting complex is hanging off the bottom. We'll talk about that more later.
Can you go again through after the electrons are passed on to quinone, like the QA and QB? I kind of got lost there.
So QA, QB, and plastoquinone are all three names of essentially the same molecule. The only real difference is the oxidation state. All right. This can receive 2 electrons, this can receive 1, and this has already received 2 electrons. So plastoquinone doesn't depart until it's picked up 2 electrons.
And D2 is only present for QA, and D1 is only present in QB.
Say that again?
D2 is only present in QA, and D1 is only present in QB.
No.
Okay.
So think of this is quinone and semiquinone. So think of these 2 molecules as something, small molecules that can diffuse around the reaction center and also throughout the lipid membrane. So they're essentially transport proteins. And the difference between QA and QB is simply the oxidation state of the quinone. And so usually when we talk about quinone, we talk about the fully reduced version. And so QA and QB is simply an easy way to differentiate between those redox states.
Okay.
Okay. All right. Plastoquinone leaves. So the one thing that I haven't mentioned here is that when we get QA and QB picking up protons, it's going to pick up the protons from the stroma. So it's going to increase the pH of the stroma, and these protons during electron transport are going to get deposited in the lumen, as we'll see in a minute.
What gets deposited in the loop?
Excuse me?
What gets deposited in the loop?
Protons. All right. So we're just all we're doing now is looking at electron flow. We've developed a 2 electron acceptor, and we're looking at proton movement from the stroma to the lumen and the ways this occurs. So one of the interesting things about herbicides, nearly all herbicides target photosynthesis. Most of those herbicides target electron flow. and by far one of the most common herbicides that's used because it's A, dirt cheap, and B, super effective at killing stuff, is DCMU. DCMU simply interacts with D1, which holds our reaction center, and prevents semiquinone from binding. In this manner, it completely stops electron transport. And usually what happens is the reaction center continues to absorb electrons. What do you think happens to it? We've sprayed DCMU on, where'd the DEA? We've sprayed DCMU on your crop. All right. What sort of phenotype do you expect to see in a week?
A dead one?
A dead one, yes. Okay, before it dies, what do we see, Melanie?
You'll see a conversion from chloroplast to etioplast.
You'll see a conversion
It won't
from
be able-
functional chloroplasts to dead ones.
Okay.
Not even etioplasts are very, very vibrant plastid organelles. We get dead chloroplasts because all of that light energy is being funneled into the reaction center, and now it no longer has any place to go. So some of it fluoresces, some of it's lost as heat, and some of it goes directly into the reaction center and just tears it up. So photosynthesis stops. What's the next thing that happens? Sugar transport stops, and it's going to start turning over all of its reserves and use all of them up. And at the end of a week, Jacob's $6 million crop is so much dust.
Is there a way to go back after the herbicide's been administered?
So some herbicides are competitive inhibitors and some are non-competitive inhibitors. Have you taken biochemistry?
I also took plant chem.
Okay. DCMU is a non-competitive inhibitor. There's no way of going back. And that's what makes it an awesome herbicide. All right. We talked about water splitting, and I don't have a slide for water splitting. The key thing with water splitting. All right, this one's not going to work very well.
Photosynthesis in what things?
Sugar export stops and uses a
Sugar exports.
All right, the most important thing with water splitting is that it requires 4 electrons to split water and generate, to split 2 molecules of water to generate 1 oxygen atom. And because all of this is happening at the molecular level, you can't split water and generate half an oxygen molecule. You have to do it, it's either all or none. Okay. And the manganese cofactor in our reaction center, there are 4 of them. So what's going to happen is that we're going to shuttle in electrons into our water splitting molecule, and in order to split 2 molecules of water, all 4 electrons that the 2 molecules of water receive must occur simultaneously. But one of the very interesting things about water splitting is that manganese has a very interesting NMR signature. And it's easy to detect the spin states of manganese. Manganese is a pretty awesome metal. It can exist +1, sorry, no, not +1, +2, +3, +4, +5, and +7. And typically what happens in water splitting is it alternates between the +2 and the +3 state. So we got four of these manganese cofactors. And chemists are able to monitor the reaction by NMR in real time. And what they observe is that the electrons actually get added sequentially, but the sequence is on the femtoscale, second frame, 10 to the minus, about 10 to the minus 15 seconds per electron addition. And this is fast enough that our free radicals don't interact with anything else. All right. Each electron and this is the kind of rate at which water splitting must work in order for it to be effective.
What does FS stand for?
Femtosecond. And this will be 10 to the minus 17. And I don't know what's more amazing, the fact that the electrons get transferred this fast, or the fact that chemists can measure reactions on that scale. I mean, both I think are pretty amazing.